chapter 3, part 6. So we're moving on to different, the rest of the parts of the cell and some other tidbits. So inclusion bodies and microcompartments. These are used for food storage, to pack gas into vesicles for buoyancy, and to store crystals of iron oxide with magnetic properties. Bacterial microcompartments, they are outer shells made of proteins and they're arranged geometrically, and they can be packed full of enzymes and designed to work together in biochemical pathways. All right, so the cytoskeleton, some bacteria produce long polymers of protein similar to eukaryotic cells for the cytoskeleton. These are arranged in helical ribbons around the cell, and they help to contribute to the cell shape. They also have found that archaea have cytoskeletons, and the cytoskeletons are unique to non-karyotic cells, and maybe a potential target for antibiotic development. So if it's found in non-eukaryotic cells, so karyotic cells other than eukaryotes, then that means then we can develop antibiotic treatment that can destroy these cytoskeletons and not have a negative effect on our cells, which is, the point of it all. Endospores. So these are dormant bodies, not metabolically active. Bacillus, Clostridium, Sporosarcina, have the ability to make endospores. So it's basically like, a really heavy-duty body of armor that these types of cells, these types of bacteria are able to make for themselves. If they are a vegetative cell, which is when they are not in the dormant state, the vegetative cell is metabolically active. Sporulation is actually induced by environmental conditions. So if something is in the dormant state or the dormant body, it's usually because the cell has been put in unfavorable conditions, such as heat, drying, freezing, radiation, chemicals. This dormant body, this endospore, will protect it. Once the cell finds itself in a favorable location where it can live without having this heavy-duty body of armor on it, then sporulation will occur. So like I said, sporulation is induced by favorable environmental conditions. And I think of endospores, and don't get me wrong, I'm just kind of using real life examples to kind of give you the idea of what these things are. I think of an endospore as kind of like a seed, because like you buy a pack of seeds and you can throw it in your car and leave it in there for 2 weeks and nothing big deal. It's not gonna hurt it. You can freeze it. You can, seeds are dried out, but you plant that seed and put some water down with it, give it some sunshine, and it likes it, and it's going to start to bud. Or if you take that seed and you put it in some damp paper towels and put it on top of your refrigerator where it's just a little warmer up there than other places, which is why my cat always likes to go up there and drive me nuts. But then that seed will start to sprout. You might, if you want to plant seeds, then you may, you may do this. You may, put them in a paper towel and get them, let them sit in a warmer area for a while, and you uncover that, like 2 days later, and they're going to be sprouting. It's the same idea with endospores, though. We don't want those to sprout, though. They're not going to sprout like a seed. Clarify, I kind of went on a tangent about seeds. Seeds are not endospores. I was just using that as an example to kind of relate to kind of how I think of it. But some bacteria have the ability to make these endospores, specifically Bacillus, Clostridium, and Sporosarcina. In your book, it has this illustration here, or this diagram of the sporulation process in the Bacillus species. So I would just kind of look over that a little bit. I'm not necessarily going to ask you for every step, but I may ask you questions related to the sporulation process. And also, it's nice to kind of see how it happens because, who knows when you might encounter this bacteria or these bacteria. Um, Bacterial endospores, medical significance of them. Bacillus anthracis has the ability to make endospores. It is the agent of anthrax. Clostridium tetani, tetanus, causes tetanus. Clostridium perfringens causes gangrene. Clostridium botulinum causes botulism. Never a good day when you get that. Clostridoides difficile, which I don't know. I feel like that might be a typo because I've usually encountered. Oh, you know what? They have changed the name. I think they did change the name. Anyway, I've always called it Clostridium difficile. You may know it as Clostridium difficile if you work in the field, if you work in a nursing home. I'm sure you've heard of C. diff. Maybe if you have an older relative or a relative that has to be on long-term antibiotics, they may have experienced a bout of C. diff. It's a serious gastrointestinal disease. Many times it occurs because someone has to be on a few rounds of antibiotics. Usually it'll occur in someone that is immunocompromised. It is highly contagious and it is not affected by hand sanitizer. So if you're around somebody with C. diff, wash your hands with soap and water. And this stuff can live on your clothing and on fabric for a very long time. So this is part of the reason why it's so easily transmittable, especially in a place like a nursing home. If one person has it, usually it's going to go down the pike and hit many people in that facility. So hand washing, very important. And you don't want C. diff. It is very distinct in its scent. If you have known anybody to have it, the smell from the stool that they pass is very distinct, and you can't miss it. When you get a sample in the lab that has C. diff in it, they're being tested for C. diff. Usually, we can tell just by the smell. I mean, we still have to go through the process of testing it, but the smell is very distinct. So I'm not gonna really get into archaea. They keep kind of going on and on about archaea in this chapter, and the one thing that I would like to tell you about archaea is on this slide. These guys live in extreme conditions, extremely high or low temperatures. Sometimes we call them extremophiles. They might need extremely high salt or acid concentrations to survive. Some live on sulfur or methane, and some can live on the human body and may be capable of causing human disease, but you don't hear about them too much. Some comparison of bacteria, archaea, and eukarya. Just basic stuff here. Not going to ask a bunch of questions on this, but you might see a question here and there. Classification systems for bacteria and archaea. So we have Berge's Manual of Systematic Bacteriology. It's a comprehensive view of bacterial and archaeal relatedness, and it's based on the ribosomal RNA sequencing. Berge's Manual of Determinative Bacteriology is based entirely on phenotypic characteristics. It categorizes organisms based on their traits, commonly assayed in clinical teaching and research labs, such as shape, seen in the microscope, or metabolic capabilities, what they need to grow. So species and subspecies in bacteria and archaea. We have bacterial species, a collection of bacterial cells, all of which share an overall similar pattern of traits. They should share at least 95% of their genes as matches. Subspecies, strain, or type refers to a bacteria of the same species that have differing characteristics. So there can be, that's like E. coli, and then you have various types of E. coli. Not all of them actually cause that food poisoning, that E. coli 0157. So there's different types, different strains of E. coli, different subspecies of E. coli. Serotype is this is representatives of a species that stimulate a distinct pattern of antibody or serum response because of unique surface molecules. And we will probably not talk about anything later on in this semester on these things, but you should just be aware of the different, think of these as terminology, right? Just know the terminology here and know what these terminologies are referring to. So that is the end of chapter 3. The rest is the appendices, if you will, and we will move on to chapter 4. And if you have any questions, let me know.